Hi, happy World Food Safety Day. I would like to thank the National Environmental Health Association for giving me this opportunity to provide you with my take on how food safety favors the connected mind. My name is Govindraj Dev Kumar, and I am an assistant professor at the University of Georgia's Center for Food Safety. I'd like to start this presentation by giving you a little bit of my background and describe how I got interested in food safety research. I'll describe how our lab uses bioluminescent foodborne pathogens to study cross-transfer and cross-contamination. We'll look at how my research using these bioluminescent organisms has helped me improve as a food safety educator and a communicator, because a picture can truly speak 1,000 words. And finally, I will give you a personal anecdote to describe how food safety knowledge is a two-way street. So I am from the city of Chennai, which is in southern India. Chennai is one of India's four major metropolitan cities. Growing up in Chennai, our main concern was waterborne diseases. So we boiled our water, we filtered it, we would add chlorine to our water. Produce safety was not something that crossed my mind. In fact, this is a typical market. Actually, this one is right next to my residence back in Chennai. And as you can see, the produce was sold by local vendors. They were usually not refrigerated, and sometimes they were kept close to the ground. In fact, my first foray into food microbiology was after reading this book called Toxin. In this book, this book was about a spurious O157Het7 outbreak associated with beef. And that got me fascinated, and I wanted to become a microbiologist. So I pursued my bachelor's and master's in applied microbiology. After completing my degree, I was lucky enough to get my first job as a microbiologist in an international airline kitchen. So we would receive foods that were prepared in different countries. So these were different types of foods that were prepared under different regulations. And as you can imagine, the microbiological testing of these foods would always yield fascinating results. So working in this stint may help me understand that food safety was a global issue. I got so interested in food microbiology that I wanted to learn further. And I moved to the United States to pursue a PhD in food science and technology at Virginia Tech. While at Virginia Tech, I looked at how tomatoes could get contaminated by dust. Following my PhD, I did my first postdoc in the University of Arizona. And at the University of Arizona, I was looking at how leafy greens such as lettuce could get contaminated by foodborne pathogens. A lot of my work was performed in the desert of Arizona, in a region called the Yuma region, which is highly productive for agriculture. I did another stint as a postdoc at the University of Maryland, and I went back to tomatoes. But this time I was studying how salmonella survives on the surface of tomatoes and actually interacts with the tomato metabolome. Finally, I am at the University of Georgia as an assistant professor, and I work on various commodities, agricultural commodities that are produced in the state. Georgia is known as the peach state, so I am working on peaches and peach safety. But there are many types of produce such as tomatoes, cucumbers, bell peppers that are also produced in Georgia. And apart from these, I work with other types of foods such as meats and specialty foods from all across the world. So it is a continuous learning endeavor, and it's very exciting. One of my lab's main area of focus is to visualize cross-transfer. Now, cross-transfer can be broadly defined as a transfer of bacteria from environmental surfaces onto food surfaces, at least in a food safety context. So in our lab, we study cross-transfer based on irrigation water misuse uh, through like overhead sprinklers or splashing. We mainly focus on dust and bioaerosols as vehicles for bacterial cross transfer. Dust is particularly interesting because it's not commonly thought of as a vehicle for bacterial transfer, and dust can actually travel across vast areas, even over continents. We have also started looking at animal intrusion and started some work looking using artificial intelligence to predict when animals intrude farms. A novel way to track the transfer and travel of foodborne pathogens from one surface to the other is through the use of bioluminescence. Bioluminescence is a spectacularly beautiful phenomenon. In fact, if you see the picture to the left, you can see bioluminescence on one of the coastlines of Australia. Bioluminescence has been observed in waters across many regions of the world, and it is a byproduct of a process called quorum sensing, where bacteria communicate with each other. So, in our research, we have used some of these genes for bioluminescence production and transferred them to Salmonella. To the right, you can see Salmonella montevideo expressing luminescence. Like I said, bioluminescence is a remarkable tool to study bacterial cross transfer. The picture that you're seeing is actually of tomato blossoms that have come in contact with dust that was carrying salmonella. You can see using this imaging technology, we can look at the distribution of bacteria on the surface of the blossom. We can look at the hot spots that tend to absorb more of the bacteria. In the case of blossoms, these were tissues that contain trichomes like fine hair. We also did other food safety experiments where we studied cross transfer from different scenarios. So we would use a knife that was used to cut meat and not washed properly, and then used to cut melons. In this picture, we had seeded the meat with bioluminescent salmonella, and you can see how the bacteria has cross transferred to the surface of the melon when the knife is not washed. We have done experiments also with not only with produce but also with meat, such as poultry and beef. In fact, in the next slide, we'll show you some of the work that we have done where we demonstrate in real time the effectiveness of certain antimicrobials and antimicrobial-based sanitizers. Finally, this picture is of lettuce. So these are discs of lettuce that were cut and contact was facilitated with soil containing salmonella. We can see how the salmonella was transferred to the surfaces of these discs. This is also an excellent tool to understand how surface roughness can affect bacterial transfer. As described previously, bioluminescence is a fantastic tool to demonstrate the antimicrobial efficacy of sanitizers. In fact, we can demonstrate the antimicrobial efficacy of sanitizers in real time. And so here's a picture of a chicken leg that was treated with a lemongrass oil-based sanitizer. And this was for a duration of 30 seconds, following which we could take the picture again and we can show the reduction in salmonella on the surface of the chicken. The reduction can be discerned by the change in color or the lack of bioluminescence on the chicken leg to the right. In fact, the only spots that seem to be holding any of the salmonella are the bone. So this not only helps scientists but also stakeholders to understand some of the safety risks associated with the products that they're making, and it helps us look for novel sanitizers in real time. This is a picture of this picture to the left is of a leaf of lettuce that has been treated with a hibiscus-based extract. And you can see that you can compare the efficacy of some of these sanitizers to each other. Finally, we have also used bioluminescence as a excellent teaching tool. This was one of our papers that got published a few years ago. And in this paper, we wanted to see how the design of coring tools contributed to the cross transfer of bacteria to lettuce. So coring tool is used to remove the core from a head of lettuce. So what we did was we seeded the coring tool with bioluminescent E. coli, and then we continued to core heads of lettuce, and we could see if the tool retained the bacteria and how much of it was transferred to the surface of the lettuce. Bioluminescent images are effective in conveying our messages to a diverse audience. While this technology has applications in cell biology, veterinary, and food science, it is also an excellent teaching tool. Our images have been used by extension experts in conveying food safety concepts to growers, producers, and consumers. We regularly use our images to cross-pollinate food safety with other sciences. One of our goals is to introduce food safety research to students and professionals across the globe. Images of our research have captured the imagination of scientists of all ages. There is a universal curiosity about food safety that is irrespective of education, background, or nationality. My current stint as the president of the Indian Association of Food Protection in North America, IAFPNA, provided our team with the opportunity to connect with a diverse audience across India. Our members presented their research to university students pursuing degrees in molecular biology, microbiology, veterinary sciences, and food sciences through seminars, question and answer sessions, and workshops. Our global outreach initiative was met with universal enthusiasm. These seminars and talks got both students and researchers interested in finding homegrown solutions to tackle pressing issues in food safety. So, as previously described, our global outreach connected us with some really interesting global solutions. We came across fascinating research on a novel strategy to control the persistence of foodborne pathogens. One of the attendees of our talks, and now a collaborator, shared his team's research on novel antimicrobial compounds. The paper can be seen to the left. The compounds were isolated from bacteria Actinomyces present in fish guts. The team isolated over 40 actinobacterial strains from fish. Some of these compounds had antibacterial action and quorum sensing inhibitory action. So bacterial quorum sensing help bacteria to sense cell density. Quorum sensing is not only involved with bioluminescence, as previously described, but also in the formation of bacterial biofilms in a variety of bacteria. When these quorum sensing compounds are blocked by inhibitors or quenchers, then we are able to control bacterial behavior such as persistence and control the formation of biofilms. Hence, quorum sensing is a mechanism of bacterial communication and is important for pathogenesis and biofilm production. The team reported the efficacy of these compounds against several foodborne pathogens, and these compounds show promise as probiotics and antimicrobials. The team has conducted further research in assaying diverse fish across India for potential antibacterial metabolite producing marine actinomyces. And hence, this kind of research on quorum sensing blockers, quorum sensing quenchers, seems to have a lot of potential in reducing the persistence of foodborne pathogens. Explorative research across the globe is vital towards finding effective solutions to food safety challenges. The isolation of novel metabolites and antimicrobials can help us combat the persistence of antibiotic resistant pathogens. Homegrown solutions towards keeping food safe and fresh can help reduce our usage of renewable energy. My favorite examples include the use of copper surfaces and use of clay containers to prevent spoilage during the long-term storage of fruit. Collaborative research could help us in better preparedness in handling emerging challenges. So now I will provide you with an example of how connecting and collaborating can help us find congruities between two seemingly non-related areas of research. So I study bacterial stress responses, and to the left is an example of filamentation, a remarkably interesting strategy used by Salmonella, E. coli, and several other foodborne pathogens. Salmonella, in this instance, can form long chains of divided but unseparated cells when faced with a stressor, such as a antimicrobial or sanitizer. These long chains begin to septate or disintegrate when the stressor abates. So when the concentration of the sanitizer reduces over time, the cells actually begin to septate. You can see the picture to the right where the cell division starts to become clear. The septa formation helps the salmonella disintegrate and actually free itself from the filaments. So this is one of the several strategies Salmonella uses to dodge antimicrobial stress. So the picture to the right consists of filamentous cells and actually some regular sized cells that are caught up in between these large filamentous cells. So the filamentous cell formation here in Salmonella was induced using a fatty acid found on the surface of tomatoes. So these are fatty acids that are found in the cuticle of tomatoes, and some of these fatty acids can induce filament formation. So I presented this research to an audience in India, and after my presentation, I got an email from one of the attendees about some interesting research that he had come across. So the article is from a Indian newspaper, and in the article they describe that a research team from the Indian Institute of Science showed that Salmonella had a knack for finding its way through nooks and corners of third-order lateral roots. So in their study, what they did was they grew their tomato plants in environments of high salinity. They also used a chemical inducer to induce third order lateral root formation. So the third order lateral roots are much finer than regular root structures. And what they found was Salmonella was able to wedge its way right into the corner and get into those nooks and crannies and enter into the root system. So this was fascinating, but it was hard for me to picture how Salmonella was so directed in its approach of entry. Normally, when you observe bacterial cells under a microscope, they're tumbling around, and unless there's chemotaxis, their movement is not very directed. Well, one day when I was observing filamentous cells under the microscope, I came across this video that I'd like to share with you. So what we see is some regular bacterial cells that are wedged between really large filaments. And what we can observe is the bacteria are actually quite dedicated in trying to get out of the tight corner. They're trying to wedge themselves out of the tight corners, and their movement is very directed. So in fact, you can see they're almost trying to nudge their way out, and there, one got away. So this video helped me understand how Salmonella might strategize and find their way through lateral root structures. So truly, one Just presenting my research to a diverse audience helped me find a new way through which salmonella might find its way through tomatoes. So discovery does favor the connected mind. I would like to acknowledge the great work by our team at the Indian affiliate for the International Association for Food Protection. Also, I would like to acknowledge Ms. Jennifer Reynolds' help, who's our communication expert, in putting this together. And finally, this was put at the University of Georgia Center for Food Safety. I'd like to thank the University of Georgia and the International Association of Food Protection for providing me with this platform to connect and communicate my research. I'd also like to thank Spectral Imaging for their imager to take these awesome bioluminescent images. Finally, I would like to thank the National Environmental Health Association and Environmental Health Australia for this opportunity to present some of my research and connect with you, the audience. I would like to thank you, the audience, for your time. I hope we get the opportunity to connect in the future. Finally, happy World Food Safety Day. Safer food, better health.